In this chapter, we'll be examining the patterns of inheritance, predominantly Mendelian patterns, and in the second portion of the chapter, we'll go into extensions that are beyond Mendelian ideas. There's been a mystery of heredity since the beginning of time. Although there were no concrete explanations of how it worked, we've always been curious about how individuals look much like their parents. Before the 20th century, there were two concepts that were the basis for all of our ideas about heredity. First of all, that heredity occurs within a species, and the second is that traits are transmitted directly from parents to offspring. It used to be thought that traits were born through fluid and blended in the offspring. The paradox here, though, is if blending occurs, why don't all individuals look alike? After all, if you were the average of your parents, and your partner were the average of their parents, then you would produce the average of you, and eventually we'd all look alike. Let's consider a couple of key people before we go on to look at Mendel. There was Josef Kohlreuter in 1760. He was working with tobacco strains and producing hybrids, that is, these hybrids differed from both parents. Once they bred these hybrids, the second generation produced offspring that varied from those of the parents. This contradicts the idea of blending or direct transmission. Another key person is T.A. Knight in 1823. He was known for crossing two varieties of garden pea. Many other people worked with garden peas before Mendel. He crossed two true breeding strains and noticed that in the first generation, these strains resembled only one parent strain, but in the second generation they went back to resembling both. So these studies gave us some information that even though a gene was not expressed, it could be carried through to the next generation. It's Mendel who put the work of these men together. He chose to study pea plants also. First of all, because other research had showed that the pea hybrids could be produced. That is, we could cross two different strains and make a hybrid of them. And that many pea varieties were available in his area. And then peas are small plants, they're easy to grow, they have a relatively fast generation time, so we could see some results. Whereas if we used human beings, well, the lifespan is quite long before individuals reproduce, so it would take a long time to get the same results. The fourth reason is that peas can self-fertilize or be cross-fertilized because the male and female parts are located both inside the flower. When we look at the flower, you can see here inside these leaves that we have the male portion, the anthers, these yellow guys here, and then the female portion, the carpel with the stigma and style inside the petals. So just inside these petals, the pea plants could fertilize themselves, as in self-fertilize. However, we can also remove either the male portion or the female portion and produce a plant that cannot self-fertilize and use a paintbrush to transfer pollen from the male portion of one flower to the female portion of another flower. Mendel generally used three stages in his experimental method. First, he would produce true breeding strains for each trait that he was studying. So he would take all purple flowered plants and he would breed them together and together and together and make sure that all he ever got was purple plants. When something is pure breeding like this, we call it true breeding. He would then cross-fertilize true breeding strains that have alternate forms of the trait. So in this example, he would cross purple flowered plants with white flowered plants. And he would not only cross them one way, as in purple pollen to the white female, he would do reciprocal crosses. So he would take white pollen and put them on the purple female parts. The third thing that Mendel would do was allow the hybrid offspring to self-fertilize, or self for short. He would allow them to do this for several generations and then count the number of offspring that are showing the form of each of the traits. In this example below, we have the anthers, the male portion, being cut away from the purple flower. And then pollen is obtained from a pure breeding or true breeding white flower and applied to the female parts of the purple flower. All the progeny from this cross turned out to be purple, which means that white has disappeared. So let's look at these crosses of Mendel's in more detail. First of all, we'll examine the monohybrid crosses. This was a cross he used to study only two variations of a single trait. So we had purple or white, yellow or green. And he spent some time producing true breeding pea strains for 7 different traits. Each trait had only 2 variants, so he chose traits with only 2 choices. Here we had round versus wrinkled, or green versus yellow, or inflated, constricted, axial flowers, or terminal flowers. And then plant height, tall or short. So each of these could either be one or the other, but nothing in between. He would start out with a parent generation. The parent generation is a true breeding parent plant. Here we have purple flowered plants being crossed with white flowered plants. This is a cross fertilization, or we're crossing purple and white true breeding parents. In the F1 generation, or the first filial generation, or simply the first generation, we see that the offspring produced by crossing these 2 true breeding strains produced only purple flowers. For every trait that Mendel studied, the F1 plants resembled only one parent, so for all 7 of those different traits. This trait he referred to as the dominant trait. The alternative trait, the one that didn't show up at all, white in this case, was called recessive. These are 2 terms we want to keep track of, dominant and recessive. So recessive is the one that does not show up in the F1 generation, and dominant is the one that does. There were no plants with characteristics that were intermediate between the 2 parents, so no light purple. It was either dark purple or white. In the F1 generation, only purple. However, when we self-cross or self the F1 generation, that is, cross this purple plant with itself, the second filial generation, or the F2 generation, showed some white plants again. So somehow the trait of white had been maintained even though it disappeared in the F1 generation. When he counted the proportion of these traits, of course, because he wasn't just breeding one flower with another, he was breeding many purple flowered plants with many white flowered plants to get the F1 generation, and then selfed all of the F1 generations and counted the proportions. He found that they came in about a 3-to-1 ratio. It's not necessarily exactly 3-to-1, but fairly close. Let's say he had 1,000 offspring, then maybe 730 of them would be purple and 270 of them might be white. This is essentially a 3-to-1 ratio. So the F2 plants had a 3-to-1 ratio. That is, 3/4 of the plants had the dominant form, purple in the case we're looking at, and 1/4 of the plants had the recessive form, white in this case. And thus they have a ratio of 3-to-1, dominant to recessive, or purple to white in this case. But when he crossed some of these F2 plants to produce an F3 generation, he discovered that some of the purple plants produced only purple plants, and some of them could produce purple or white plants. And from these ratios, he decided that there was one true breeding dominant purple, in this case plant, and 2 purples that were not true breeding, and then 1 true breeding recessive plant. Now you have to keep in mind that Mendel did not know anything about how meiosis works, nor did he know of Darwin's work. So he came about these ratios all on his own, and he used them to explain basically what's going on in meiosis. His plants did not show intermediate traits. Each trait is intact and discrete and maintains itself throughout the different generations. Sometimes they appear, sometimes they don't. He also concluded that for each pair, one trait was dominant and one was recessive. That pairs of alternative traits that he examined were segregated among the progeny of a particular cross. This conclusion was very important because this means that alternative traits, as in the purple and the white, become segregated. When we say segregated, we just simply mean separated. He's basically saying that sister chromatids separate from each other. That is, genes for the same trait separate from each other somehow in the formation of gametes. And that these alternative traits were expressed in the F2 generation at a ratio of 3/4 to 1/4. That is, 3 dominant to 1 recessive. To explain his results, Mendel proposed a simple model that became one of the most famous in the history of science, containing simple assumptions and making clear predictions. The model has 5 elements. The first is that parents transmit discrete factors. Of course, he didn't know that they were called genes yet, so he called them factors. That is, purple and white maintain themselves as discrete units. They did not blend together. The second is that each individual receives 1 copy of a gene from each parent. The third is that not all copies of a gene are identical. He said there are alternative forms of genes, which he termed allele. So alternative forms of genes are alleles for a different gene. Purple is one allele and white is another. An individual that possessed 2 of the same allele was considered to be homozygous. Homo meaning the same, zygos meaning in the zygote, had the same 2 factors in the zygote. And the individual that had 2 different alleles was called heterozygous. Hetero meaning different alleles in the zygote. The fourth principle, though, is that alleles remain discrete. That is, there's no blending. So purple is there, and even though it's covered up, white could still exist. The fifth was that the presence of an allele does not guarantee its expression. Here he provides 2 new terms. There's a dominant allele, that's the one that's expressed. In the case we were just looking at, it was purple. And a recessive allele. The recessive allele is hidden by the dominant allele. Now keep in mind, he's only looking at 2 discrete traits. He picked 7 true breeding traits with 2 discrete options. Here's a couple of other terminologies that we'll need to get through the rest of this chapter. The first is genotype. Genotype is the total set of alleles an individual contains. In more clear terms, the genotype is what genes there are. Big P and little p, or big P, big P. Whereas the phenotype is the physical appearance. What does the individual actually look like? It's purple or it's white. So genotype will be big P, little p. Phenotype would be purple. Now I think we have all of the terminology that we need. But finally, let's consider what the whole experiment of monohybrid crossing was about. It's about the principle of segregation. So monohybrid cross experiments of Mendel were used to come about the principle of segregation. It's stated like this: the 2 alleles for a gene segregate during gamete formation and are rejoined at random, 1 from each parent, during fertilization. The prime part is that 2 alleles for a gene segregate. And we've seen this exhibited in the last chapter when we see homologous chromosomes separate from each other during meiosis 1. The 2 alleles, one on the chromosome from mom and one on the chromosome from dad, segregate during the formation of gametes. The physical basis for allele segregation is the behavior of chromosomes during meiosis. Again, this is pretty amazing stuff considering Mendel had no knowledge of chromosomes, he had no knowledge of meiosis, for these things had not yet been described. Pretty amazing stuff. To test his models, Mendel first expressed it in terms of simple sets of symbols. He used these symbols to interpret his results. Consider again Mendel's cross between purple flowered and white flowered plants. The P is the dominant allele. We denote this with a capital P. The recessive allele is noted with a lowercase p. It's really important here to make a big distinction between your capital P's and your lowercase p's, otherwise we can make quite a mess. So a true breeding white flowered plant then is going to be small p, small p. This is said to be homozygous recessive because it only has one type of allele and they're the recessive ones. The true breeding purpled flower plant is big P, big P. This is homozygous dominant. And that which is big P and little p is considered to be heterozygous. Remember, with heterozygous, we don't need to say dominant or recessive because it's heterozygous. This would produce a purple flowered plant in this simple dominance system. So these are the notations that we'll use to keep track of the different traits. When the F1 individuals are allowed to self-fertilize, the P, big P, and little p alleles segregate during gamete formation to produce gametes that have either big P's or little p's. And then we'll look at their subsequent union in the F2 individual. It's random. In order for us to keep this visualized, we'll use a simple diagram called a Punnett square. It's named after its originator, a geneticist from England called R.C. Punnett. Let's take a look at how the Punnett square works. So remember, we need to consider all the possibilities for gametes. In an individual that is big P and little p, as would be the F1 generation of Mendel's monohybrid cross, the sperm could either produce gametes that are big P, or we could produce gametes that are little p. I always draw circles around my gametes. The other parent, let's say the female, the egg-donating parent, could also produce eggs that have big P's or eggs that have little p's. So we can look at this random combination of what might happen on fertilization. Simply here, we take the little p from here and the little p from here to produce an offspring, which is little p and little p. We'll do the same thing. We'll now take the big P from here and the little p from here. Fill in this box with a big P and a little p. And we continue this process until we get to all the squares being filled. What we see in this final Punnett square is that we have 3 purple, because big P and little p makes purple, and so does big P and big P, and 1 white. Here we have our 3-to-1 ratio that Mendel found when he was breeding his pea plants. We'll use the Punnett square frequently from now on to follow our monohybrid and dihybrid crosses. So we may as well practice right now. Let's do a practice Punnett square. We have one parent that is big P, crossed with another parent that is big P and little p. What ratio do you get for offspring there? So a number of human traits have been shown to display both dominant and recessive inheritance. However, researchers cannot perform controlled crosses on humans in the way that Mendel did with pea plants. First of all, we have a really long generation time. And second of all, it's much more complicated. Instead, geneticists study crosses that have already been performed. In other words, we explore family histories. The organized methodology we used is called a pedigree. We can use a pedigree for traits that are controlled by a single gene because they do exhibit dominant and recessive inheritance. Pedigree analysis is used to track inheritance patterns in families. Below, you can see a dominant pedigree. An example is juvenile glaucoma. This disease causes degeneration of the optic nerve and leads to blindness in children. It's a dominant trait and it appears in every generation. That is, if one parent has it, the likelihood of a child having it is very high because there only has to be one allele for that trait to show up. Let's say we're looking at glaucoma in Mendel's notation. An individual that is big G will be affected. An individual that is big G would also be affected because it's a dominant inheritance pattern. However, an individual that is little g would not be affected. This is the recessive condition. So there would be a 3-to-1 possibility that a child would have glaucoma. Because of this frequency, a dominant trait is likely to appear in every single generation. We can see it in this generation, and in these generations. Now, a quick note about pedigrees. We note the generations with Roman numerals and the individuals in the generation with Arabic numerals. An affected individual has a shaded symbol, and unaffected individuals have unshaded symbols. Circles will always represent females, and squares will always represent males. If we know that an individual is heterozygous, we might have half of the squares shaded. Let's look at a recessive pedigree to contrast. Albinism is a condition in which pigment melanin is not produced, and thus an individual doesn't have any pigment in their hair or skin. The pedigree for albinism. One form of albinism is due to a non-functional allele that creates the enzyme tyrosinase, which means the pigment cannot be made. Males and females are affected equally, and most affected individuals have unaffected parents because it's a recessive trait. When we look into generation 3, we can see that individuals have albinism. That's because 2 of the parents must have been carriers. In this case, we could have a possibility of their progeny being either carriers or fully affected with albinism. In this case, let's say we're looking at A for albinism. An individual who is big A, does not have the condition. An individual who is heterozygous, big A, little a, would not have the condition either. He would be a carrier because of this little a. An individual with little a is the only individual that will actually be affected. So most often, recessive conditions skip generations. Here's a number of different dominant and recessive traits. Play with a few of these and do a few Punnett squares and see how many individuals are affected versus not. Perhaps even draw a pedigree or two. Let's take a quick card quiz. In a cross between a homozygous purple flowered plant and a homozygous white flowered plant, all of the F1 hybrid offspring have purple flowers. What is the term for the white trait? In heterozygous individuals, there are blank alleles. The principle of segregation states, a diagram of genetic relationships among family members is termed, when creating a Punnett square, it's assumed that all possible types of sperm fertilize all possible types of eggs. Is this true or false? Here are your answers. If you got all of these, you're well on your way to understanding the monohybrid crosses. Please practice some monohybrid crosses. Again, use some of these traits that we see in this table here. Create some symbols. Albinism can be big A and little a. Hemophilia, big H and little h. Try it out. See what you come up with.